Vaccines and the immune response. How vaccines work. Influenza vaccines are able to trigger an immune response by mimicking viral infection. They are usually manufactured using inactivated or killed virus particles taken from various circulating influenza strains. Inactive fractioned viral components contain the subvirion particles hemagglutinin, HA, and neuraminidase, NA. In Canada, influenza vaccines are administered via intramuscular injection. These fractioned particles containing the foreign antigens hemagglutinin and neuraminidase are released from the vaccine into the bloodstream. There, they are met with an immune response mediated by various immune cells, including macrophages, T lymphocytes or T cells, and B lymphocytes or B cells. A major mechanism of action involves macrophage phagocytosis of hemagglutinin, breaking it into smaller components. After ingestion, macrophages display the hemagglutinin antigen on their surface in combination with a specific receptor known as the major histocompatibility complex or MHC. T cells are now able to recognize and bind foreign antigens that are associated with the MHC. Upon binding to the MHC receptor, T cells become activated and proliferate into either cytotoxic T cells, regulatory suppressor T cells, or helper T cells. Activated helper T cells express hemagglutinin receptors specific to the vaccine strand on their surface and play a major role in antibody generation and memory B cell activation. Unlike T cells, B cells are able to ingest hemagglutinin independent of the MHC. Once internalized, B cells process the hemagglutinin antigen and present it on their surface in combination with an MHC. When activated helper T cells interact with activated B cells expressing antigen MHC receptors, they begin secreting lymphokines which have several effects. Lymphokines trigger activated B cell proliferation, which leads to either their differentiation into memory B cells or into plasma cells. Plasma cells produce hemagglutinin antibodies specific to the strain of influenza contained in the vaccine. Memory B cells aid in future immune response when exposed to an active influenza virus. When an infected host sneezes towards an uninfected person, the nasopharynx is exposed to aerosol droplets containing whole, live influenza virus. Once inhaled, the influenza virus attempts viral colonization of nasopharyngeal epithelial cells. Assuming that the vaccine strain matches that of the live virus, hemagglutinin antibodies block viral attachment of live influenza virus to host epithelial cells, and overall disease is either avoided or diminished in severity.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1 which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.